And now we go back to the vaccine approach. So our next speaker is Dr. Harman Mali. Immunization with a self-assembling nanoparticle vaccine displaying EBV GHGL protects humanized mice against lethal viral challenge.
Yeah. Not a doctor yet, but hopefully soon.
Okay.
So just thank you to the organizers for giving me a chance to share my research here today. My name is Harman Mali, and I'm a fourth-year graduate student in the McGuire Lab at Fred Hutch. And today I'm going to be sharing my thesis project, designing a next-generation Epstein-Barr virus vaccine. EBV is orally transmitted through saliva and is capable of infecting both epithelial cells and B cells. It's not clear what immune response a protective vaccine would need to elicit, but given that the correlate of protection for most licensed viral vaccines out there are neutralizing antibodies, it's likely that an EBV vaccine would need to elicit neutralizing antibody against infection of both of these cell types. When it comes to designing a vaccine, obviously the most important thing you want to consider is which antigens to include. And a logical first step there is to focus on proteins that are involved in viral entry. The idea being that if you can prevent entry, then you can prevent infection from occurring in the first place. With EBV, there are several proteins involved in entry. Other speakers have already gone over them, so I'm not going to re-summarize that, but I do want to reiterate the point that most of these proteins that are involved in entry are actually targeted by neutralizing antibodies during the course of natural infection, and we believe that any of them could potentially be included in a EBV vaccine. In the past, most EBV vaccine efforts have focused on GP350. However, it has been suggested in the literature that the inclusion of other proteins might lead to a more protective vaccine. Our lab wanted to further explore which other proteins might be important for an EBV vaccine. So we set about trying to isolate monoclonal antibodies against EBV proteins in EBV-infected individuals. And through that, we were able to isolate AMA1. Unlike the other anti-GHGL monoclonal antibodies, which had been previously isolated, shown here, AMA1 was capable of potently neutralizing both epithelial cell infection as well as B cell infection, which suggested to us that if you could elicit AMA1-like antibodies through vaccination with GHGL, it might be an important EBV protein to include in a vaccine. Since then, the discovery of other potent anti-GHGL monoclonal antibodies, such as 769B10, which Jeff Cohen shared earlier, as well as and 1D8, which targets the distal epitope to AMA1's epitope, discovered by the Zhang lab. The discovery of these further supports the notion that GHGL might be an important target for an EBV vaccine. Our lab has shown that the passive transfer of AMA1 to rhesus monkeys protects them against challenge with RHLCV, which is the rhesus ortholog of EBV. Likewise, our lab has shown that passive transfer of AMA1 to humanized mice protects them against challenge with human EBV, and that was the case in 11 out of 13 mice. 1D8, the other anti-GHGL monoclonal antibody I shared earlier, has also been shown to be protective against EBV challenge in humanized mice. Taken together, these studies really provide a proof of concept that anti-GHGL antibodies, specifically AMA1-like antibodies, may be protective in vivo against infection by EBV, and that if you were able to elicit AMA1-like antibodies through GHGL vaccination, perhaps that vaccine too would be protective. Antibodies elicited by that vaccine could also, confer protection. Several researchers have shown that through multimerization, you can improve the immunogenicity of protein subunit vaccines. We wanted to explore that further in the context of GHGL, and we did that using a combination of both computationally designed as well as naturally occurring protein subunits. The former that we used was designed by Phil Bradley at Fred Hutch. The 7-mer we used was derived from the C4b binding protein from Gallus gallus. The 24-mer we use is the same H. pylori bullfrog ferritin used by the Cohen lab. And the 6-teamer that we used was designed by Neil King's lab at the University of Washington. We fused GHCl to each of these multimeric protein subunit backbones, and in order to ensure that they express with the expected structure, size, and antigenicity, we proceeded to characterize them. One of the ways we did this was by collecting electron micrographs of each of these constructs. And as you can see here, we were able to confirm that each of these multimers expressed as monodispersed particles with no aggregates or any sort of clumping forming. And as you might expect, with the increasing valency, we saw increasing size in each of these constructs. So they're expressing about as we would expect. The next thing we looked at was antigenicity using a panel of anti-GHCl monoclonal antibodies. And in each case, we saw that each of these constructs retained their antigenicity, and in most cases, multimerization actually improved the antigenicity of GHCl. Having confirmed the expected antigenicity, structure, and size of each of our constructs, we proceeded to immunize C57 black 6 mice with our constructs. We performed 3 immunizations and collected blood after each immunization in order to measure plasma neutralizing titers against EBV infection of both epithelial cells as well as B cells. I'm going to go over the epithelial cell neutralizing titers first, as measured here by reciprocal ID50s. The way to interpret this is, the higher the potency of the neutralizing activity, the higher value you'll get with the reciprocal ID50. And as you can see here, after the first immunization, you already begin to see significant differences between the monomer, where there was no measurable neutralizing activity, and the multimers, where you saw titers sometimes past 10 to the third. Post-second, you really see the clearest differences between each of our multimer constructs and the monomer here. You start to see some neutralization. However, neut titers are as much as 2 log-fold higher, and in each case, significantly higher in each of the multimers. By the third time point after that third immunization, we see a further boost in titers with the monomer, and titers sort of begin to converge across each of these constructs. The next set of data are the B cell neutralizing titers. These titers, it's worth pointing out, they're more difficult to elicit, so we do see them show up later and at relatively lower values compared to the epithelial cell neuts. However, the trend of the data is pretty similar. Post-second, we see the clearest differences between the monomer, where there's really no neutralizing activity, and each of the multimers. And again, post-third, these begin to converge. We next wanted to sort of explore the relevance of these clear differences we see in vitro between monomer and multimer. We wanted to explore that in vivo, the relevance of that in vivo, and we chose to do that using the 6-teamer, which, as you can see here, has consistently high neutralizing titers in both of these assays, and we want to compare that with the monomer. And we did so using a passive transfer approach. We, in humanized mice specifically, we did this in immunocompromised mice engrafted with human CD34 stem cells. We chose that approach over direct immunization because, due to the fact that humanized mice are immunocompromised, they don't mount a very strong antibody response. And this experiment, it took place in 2 parts. In the first part, we generated that vaccine-elicited IgG by immunizing large groups of mice with either a GHCl 6-teamer or monomer. We then purified IgG from the blood of those mice. We also included a non-vaccinated group to control for the passive transfer, which took place in part 2. In part 2, we transferred 500 micrograms of the vaccine-elicited IgG to each humanized mouse. At day -1, we then bled the mice to ensure that each mouse received roughly equivalent levels of total IgG. And then at day 0, we challenged them with a high dose of EBV. And we included an infected control there, which had no treatment or any transfer, as well as an uninfected control. We then monitored those mice for 10 weeks for any signs of infection. The first sort of metric of infection that we looked at was survival. In the infected control group, the mice didn't survive past day 60. So in all 5 of the mice succumbed to the lethal challenge, whereas the uninfected control, as you might imagine, survived through the length of the experiment. The next group I want to look at is the control IgG. These mice received IgG, but it wasn't any vaccine-elicited IgG, and they also received challenge, and they fared about as well as the infected control, as you would imagine. Now, the mice that received the monomer vaccine-elicited IgG, they fared a little bit better than the infected control groups. 1 of the 4 mice did survive through the length of the experiment. However, by contrast, all 5 out of 5 of the, it didn't show here, but this purple line here, all 5 of the mice in the 6-teamer group were protected against lethal challenge. So already you begin to see sort of the superiority of vaccine-elicited 6-teamer IgG compared to the monomer vaccine-elicited IgG in terms of protection against challenge. The next measure we looked at were DNA copies in the blood, or viremia. As we saw with the infected control group, we saw viremia as early as day 21, and by day 49, each mouse had detectable levels of viremia in the blood, and as you expect in the uninfected control group, there's no detectable viremia through the length of the experiment. And it's worth pointing out, this agrees quite well with the survival data where we saw none of the infected control mice survive through the course of the experiment. As with the survival, the control IgG group mirrors quite well what we saw with the infected control group. With the monomer IgG, the 3 mice that didn't survive the challenge had high levels of viremia. However, that 4th mouse that survived, while it did survive the lethal challenge, it did have detectable viremia at day 70. And then finally, the 6-teamer group, while all the mice did survive, at day 70, we did see 3 out of those 5 mice have detectable levels of viremia. However, compared to the infected control groups, this was delayed really until the final weeks of the study. The final measure that we looked at was EBV DNA and splenic extracts. I want to point out that the 2 mice that remained EBV negative in that 6-teamer group, one of them, we did have detectable levels of EBV transcript in the spleen. However, it was just above the limit of detection. And we did see a significant reduction in the levels of EBV DNA in the spleens of the 6-teamer group, despite having that positivity. So in summary, multimeric GHCl constructs, they elicited superior neutralizing titers than monomeric GHCl in vitro. And we also showed that passive transfer of IgG elicited by the GHCl 6-teamer, 1, provided superior protection against lethal challenge than IgG elicited by the GHCl monomer, and 2, delayed viremia in those humanized mice. And thinking towards the future, we really believe that immunization with multimeric GHCl is a viable strategy for eliciting neutralizing antibodies against EBV infection and may be important for designing an effective EBV vaccine. Finally, just want to thank everybody in my lab, as well as our various collaborators across these various Seattle institutions. And thank you all for listening.
Thank you. And it's open for questions. Please.
Hi, great talk. I'm wondering with your 6-teamer, if it's large enough to add other proteins that are involved in attachment and fusion, if you think that would be worth doing to improve reducing viremia past 70 days in those mice.
Yeah, so we do think that it would be important, perhaps a complex GHL with other important antigens such as GP350. As Jeff mentioned during his lecture, other groups such as Moderna and other labs are also doing that. It's probably going to be important in, if you're targeting more antigens, it would make sense that that might be more protective. In terms of whether or not it would delay viremia, we won't know until we actually do those studies. But I think, it's an important thing to look into, and it's worth doing. Yeah.
Please.
Could you comment a little bit more about the scaffold for the 6-dimer? Is that a sort of an artificial protein? Do you know whether antibodies are made to that could affect things? Is it a single protein or multiple proteins for that 6-dimer?
Yeah, so for that, it's a single protein. It's I3. I believe it's either in preprint or it's been published by the King Lab. We looked into whether or not we generated through those vaccinations any antibodies against that, and we weren't able to detect it. I don't think we've published that, or it's not in the paper. But we looked at that. We also looked at MDT, and we didn't really see any titers against the I3. But it is possible that you could generate titers against those backbones. So it's something worth looking into.
I have a couple of questions myself. Oh, sorry.
Yeah, quick question about the dosing. For the comparison in animals, and I'm sorry if I missed it, but what was the dosing? Was it based on GHGL or was it based on total protein?
Yeah, so that was based on total protein. It was 5 micrograms, and that was adjuvanted with, in the original set of data where I showed the neutralizing titers, that was with SLA-SE from IDRI. However, when we did the humanized mouse study, we used SAS or RIBI, and we compared the titers, and they were equivalent between those two adjuvants. They're both, lipid-based adjuvants, so it's about what you would expect. But yes, so it's total protein.
When you were looking at the neutralizing antibodies produced by the various multimers, the 4, 7, 24, 60, it looked like they were all better than the monomer. So I'm just wondering why you chose to go with the 60 for your further studies.
Yeah, so I think part of it was just the limitation of, how many mice you'd have in the study. It was really the 6-dimer, it was consistently quite high. There were other groups that we could have chosen, and I think if we could have used all of them, we would have. But that's the one that we elected to go with.
Okay, my questions were about, if it's done there, okay. About, when you compare this monomer versus multimer, you just mentioned now that you used couple of different adjuvants in different experiments, but they were all the same kind of type of adjuvant coming from IDRI?
Mm-hmm.
Okay. So question is, have you thought about playing with different adjuvants of a different nature with the monomer and maybe obtaining titers that you would compare, when you take, the trouble with your 6-dimer, okay? First question. Second question, when you talk about superior neutralization titers, okay? The superior neutralization titer, you imply also the quality of the antibody response, say, for instance, in the total avidity of the sera. Have you tested anything of this type?
Yeah, so maybe this might be answering, so another thing that we looked at were binding titers. So in addition to sort of having those higher neutralizing titers, we also looked at binding titers, and it's similar to what we saw in terms of the trend with the neutralizing titers, where it was higher with the multimers. And that, the title that I have there, we've published that data in that paper, and you can see we do see those differences there. In terms of looking at other adjuvants, I do think that would be really interesting. There's a matrix M, for one, that Cohen Enable, and that sort of group is using for the GP350 vaccine. I think saponin-based adjuvants also have, a lot of they're really great at adjuvanting or immune response. And there's obviously lots of other adjuvants out there that you can look at as well. And I think it's worth doing, definitely.
Okay, thank you so much. Any other questions from the floor? Okay, we go to the next speaker.